Good afternoon, ladies and gentlemen. My name is Konstantinos Mitsakakis. Unfortunately, I could not be with you live due to a family emergency, but it is my pleasure to present our work on integration of a bead-based immunoassay on a commercial PCR-performing point-of-care device. The background of our work is around the detection of host response biomarkers in the context of infectious disease diagnostics. It is known that pathogen identification through nucleic acid amplification, for example, PCR or isothermal amplification, is key for evidence-based diagnosis and patient management. However, nucleic acid amplification alone may not be able to differentiate colonization versus infection, or may not be able to differentiate cases of sepsis versus non-sepsis inflammations, or may not be able to define disease severity. In such cases, the quantification of host response biomarkers for complementary assessment to pathogen identification can improve diagnostic accuracy and efficiency. However, there is one main challenge. The procedures of pathogen identification and biomarker quantification require different methods, nucleic acid amplification and immunoassay respectively. This means that different instruments are required, which increases the overall cost. For example, a hospital would need to buy different instruments, and the different methods may require different settings for analysis, for example, microbiology versus clinical chemistry departments, which may likely cause delays of the whole process for patient management. Therefore, the proposed solution and goal of this work is to develop an immunoassay that is compatible with and integrable in point-of-care nucleic acid amplification instruments. We used CRP, C-reactive protein, as a case study due to its utility in guiding antibiotic prescriptions. For example, in the UK and the Netherlands guidelines for respiratory tract infections, CRP less than 20 mg per liter means that typically no antibiotic prescription is done. CRP between 20 and 100 means that the clinician needs to co-assess with other clinical parameters such as oxygen saturation, fever, etc. And CRP higher than 100 mg per liter means that antibiotic prescription is recommended. The immunoassay that we developed for the scope of our work is named Bound-Free Phase Detection Immunoassay, in short, BFPD. The competitive format of BFPD is shown in this figure. In particular, the assay consists of magnetic beads, 2.8 micrometers in diameter, coupled with a capture antibody, fluorescent beads, 200 nanometer in diameter, coupled with a competitive antigen, the sample itself with the analyte of interest, and one assay buffer. These components are mixed, and after a single incubation step, the magnetic beads with whatever is bound in them are separated, for example, using external magnets in the case of a microtiter plate. Then a portion of the remaining unbound beads, fluorescent beads, is transferred to another reaction well, and fluorescence is measured in this bound-free phase. The assay performance was first assessed in a microtiter plate, followed by readout on a standard Tecan fluorescence reader. We acquired a standard curve that covers the clinically relevant cutoffs of 20 and 100 mg per liter that we saw before. The incubation time was 15 minutes with sample volume, that was serum, of 5 microliters. We performed a method verification using an EU certified reference material, which is serum with a well-defined CRP concentration measured using immunoturbidimetry. The expected value was 41.2, and we measured over 5 different days, and our measured value using the BFPD was 40.5 mg per liter, which was pretty good. Furthermore, we tested 37 clinical serum samples where the CRP concentration was quantified by BFPD, ELISA, and immunoturbidimetric assay, and the correlations between the methods were BFPD versus immunoturbidimetry was 0.95, and BFPD versus ELISA was 1.12, which is also pretty good, considering that the two reference methods between them had a correlation of 1.15. So in the beginning we said that our goal is to integrate an immunoassay in a PCR-performing instrument. For this reason, we chose a centrifugal microfluidics and the lab-disc technology because centrifugal microfluidics do not require external pumps or tubes, and because the liquids can be handled through the interplay of capillary, centrifugal, and thermopneumatic principles. The corresponding lab displayer functional model device that we see contains a thermal unit, a fluorescence readout unit, and built-in magnets for handling magnetic particles. The lab displayer had already been proven able to process PCR discs for applications like fever diagnostics, oral bacteria detection, and respiratory pathogen detection. And the question is the same device capable to detect CRP? The answer is yes, through the immunodisc cartridge that you see in this picture, and that contains 3 identical structures enabling the analysis of 3 samples simultaneously on the same disc. The immunodisc is a centrifugal microfluidic cartridge with fully integrated reagents for performing sample-to-answer analysis. In particular, the assay buffer is stored in aluminum pouches called stick packs, like the one you see in position 2. The protein-coupled beads were dry stored, as you see in positions 3 and 4. We performed a parametric study to define the drying buffer compositions, as well as the drying temperature and duration, which had to be the same for the fluorescent and magnetic beads, as they are both stored in the same cartridge and need to be dried simultaneously. More details on the parametric study can be found on the relevant publication. And the steps of running the immunodisc are in general the following. In position 1, we insert the serum in the inlet. Then, during rotating, the stick pack in position 2 burst opens and releases the assay buffer. Then the liquid mix, so the assay buffer goes to chamber 1, and the liquid mix goes to chamber 3 and dehydrates the fluorescent beads. Then the liquid mix goes to chamber 4, rehydrates the magnetic beads, then incubation starts, the separation follows, and finally the detection is done, and all these in one chamber. We performed a demonstration of the immunodisc using CRP with 5 microliters serum in 75 microliters total volume and achieving 20 minutes time to answer. We had a quantification range from 18.2 to 115, covering the cutoff values of 20 and 100 that we saw before. And regarding reproducibility, we achieved an inter-day signal CV of the standards of 5.8%. We also measured the CRP certified reference material using 3 batches of discs. The expected value was 41.2 and we measured 46.9. The discrepancy is anyway small, but we attribute it to the different principles of measurement methods, immunoturbidimetry versus our BFPD immunoassay, and also to the fact that we had dry pre-stored reagents. Importantly, the interbatch CV of the measured value of the CRM was less than 10% and in particular 8.6%. So we saw until now that the BFPD immunoassay can be performed on a lab disc player functional model instrument that also performs the PCR discs. The question is, how about a real commercial instrument? Spindiak GmbH is a company that commercializes the Ronda system. It is a device that processes half discs for detecting SARS-CoV-2, RSV, and influenza viruses using rapid PCR and starting from a swab that is directly inserted into the disc. So the goal was to integrate the BFPD immunoassay on this commercial PCR-performing instrument where little to no changes in its hardware are possible, and taking into account the special requirement for increased throughput in analysis during the COVID-19 pandemic. The heart of the BFPD immunoassay fluidics is the multi-purpose chamber. We can see a picture, a design here, in which incubation, mixing, separation of magnetic beads and detection of the bound-free fluorescent beads are all performed in a single chamber. In the photo here, we see the design of this chamber, and on the left and on the right, we see a real photo where we observe the sedimented magnetic beads, in this case using centrifugal forces, and the dispersed or bound-free fluorescent beads here, as well as the detection area. Then, the addition of 7 such multi-purpose chambers in a half disc gives us the Ronda immunodisk, where we can see the half-disk A and the half-disk B, which can both run simultaneously on the SpinDiag Ronda device. And on one hand, we can say that we have less degree of integration in this half-disk, as we have pre-stored only the magnetic beads, and we pipette in the inlet the fluorescent beads, the sample, and the assay buffer. But on the other hand, we do achieve an increased degree of throughput on a point-of-care instrument, since we can acquire 2 times 7, that is 14 data points per run. It is also worth mentioning that the reproducibility between chambers was calculated to be 5.4%, which is also pretty good. This Ronda immunodisk was demonstrated using COPD, that is chronic obstructive pulmonary disease, as a clinical scenario. In this scenario, and according to studies, there are 2 clinical cutoff values of interest, the 20 and the 40 milligram per liter, which create 3 concentration zones. Zone A, where CRP is below 20 milligram per liter and no antibiotics are recommended. Zone B, where CRP is between 20 and 40, which is a gray zone and needs co-assessment with other health parameters. And the Zone 3, where CRP is higher than 40 milligram per liter, in which case the antibiotic prescription is recommended. Using the Ronda immunodisk, we calculated the limit of detection to be 2.3 milligram per liter, and we tested 27 clinical serum samples using the BFPD on the microtiter plate, as well as the BFPD on the Ronda immunodisk. The outcome was that the 2 methods agreed in 22 out of 27 cases, the green marked here, in terms of classifying the samples in the same concentration zones, A, B, or C. Of course, with room for further improvement, we nevertheless consider this a very satisfactory result, since we managed to test real samples with a new immunoassay method on an automated commercial and primarily PCR-intended instrument. Conclusively, the BFPD is an immunoassay method that includes only few steps and a simple workflow, which allows for easy integration, is wash-free, which allows for saving material and therefore costs, and requires no sample pre-dilution before the assay itself, which offers the advantage of reducing errors. The BFPD immunoassay is an assay platform itself and was demonstrated on 2 PCR-performing instruments, the LabDisplayer 1 functional model through full integration and spike samples, and the SpinDiag Ronda commercial instrument with increased throughput and real samples. Future plans for integration in further amplification-performing devices includes the currently running EU project HolyCare, where BFPD integration is done on a non-centrifugal system, and the currently running national BMBF project Diagnoseps, where BFPD integration is done on a robotic station. We also plan to expand the BFPD biomarker portfolio towards procalcitonin and interleukin-6 for infectious diseases, as well as other biomarkers for chronic non-infectious diseases. And reaching the end of the presentation, I would like to thank, and on behalf of all the co-authors, our colleagues at Hahn-Schickard, the Hahn-Schickard Foundry Service for the LabDisc cartridge production, our external project partners, the funding agencies and projects, Horizon 2020 Diagoras, Eurostars, Respiotic, BMBF, Septicision, and Covires, and of course you for your attention. I would also like to thank very much the organizers for the very kind opportunity to allow me to present our work in a recorded instead of a live manner. And please feel free to contact me in this email in case you'd like to follow up on our work. And finally, I wish you a fruitful remaining Eurosensors conference. Thank you very much.